Aloha everyone, and welcome to our third and final lecture on Chapter 3 in your textbook, which was all about cells. So today we're going to be talking about protein synthesis. So today we're going to be wrapping up our discussion of cells, which again is Chapter 3 in your textbook. We are going to look at our key questions and learning outcomes. Then our lecture will be covering the process of protein synthesis. And then after you finish watching this lecture, you have a transcription and translation activity for this week to do. If you want, I would encourage you to kind of pull that up on your computer or print it off before you watch this lecture. And you should be able to fill in most of that while watching the lecture, kind of like taking notes and doing an assignment at the same time. So I kind of purposely did that to kind of make the best use of your time. Or if you prefer, you can do that after you watch the lecture as kind of a review, make sure you understand everything. So your choice there. And then you also will have a quiz for this week to take over Chapter 3. And because this is our last lecture on Chapter 3, that's why I put this as something to do. You don't have to do it today, but just by the end of the week, completing that quiz on Chapter 3. So again, today we're going to answer these key questions. We're going to look at why is DNA transcribed and how do genes relate to specific proteins. So in the last lecture, that 3B lecture, we talked about the cell cycle and mitosis, right? We talked about DNA replication there. And now today we're going to talk about one more process. We'll be talking about protein synthesis, which includes transcription and translation. In terms of our learning outcomes for today, we're going to talk about the genetic code. We're going to talk about transcription and translation, so defining those different terms. We're going to talk about making materials from DNA via a sequence of events, specifically translation and transcription. We're going to talk about and explain the process of RNA synthesis. And then we're going to basically talk about how we have all of these key players in protein synthesis. So we're going to talk about our mRNA codons. We'll talk about tRNA anticodons. We'll talk about DNA polymerase. I already mentioned that a little bit before with DNA replication. We're going to talk about promoters. We're going to talk about ribosomes, proteins, amino acids, and base pairs. So all of those words should kind of ring a bell, maybe from a previous class, but if they don't, that's totally okay. By the end of this lecture, the goal is to understand what those terms mean and the roles that they play, and also to be able to answer the questions that we covered on the previous slide. So let's go ahead and get started talking about protein synthesis, which is again just like what it sounds. Protein synthesis is the biological process of building proteins from those building blocks amino acids. And this is going to be in accordance with that genetic blueprint that's encoded by DNA. So we're going to need DNA for this process of protein synthesis. So this is what we're going to be doing. We're going to be making connections with these things today. So we already talked about DNA and RNA in isolation when we talked about the organic molecules in that biochemistry lecture. And then in our last lecture, we covered this process right here, which is your DNA replication, where that DNA is going to be split by that enzyme helicase, splits it. Those free-floating nitrogenous bases are going to be attracted to those open ends on that leading and lagging strand, and then we're going to have DNA polymerase actually form those two new strands using those hydrogen bonds to put those bases back together. So again, that's DNA replication. We talked about that in the previous lecture. What we're going to be doing today is looking at how we have this process of taking DNA, we're going to transcribe it into mRNA, so that's going to be this RNA synthesis, and then that mRNA is going to be translated into our proteins. So this is what we're going to be covering today, this connection between our DNA, our RNA, and our proteins in this process of protein synthesis. So first, let's go ahead and review the basics here. So the DNA in our cell is going to hold the instructions on how to make the protein, which is why every cell in the body must have a copy of the DNA. As a reminder, the DNA is the blueprint of life, right? Because it contains the instructions needed for an organism to grow, develop, survive, and reproduce. DNA does this by controlling this process of protein synthesis. Proteins are actually what do most of the work in the cells, and they are the basic unit of structure and function in the cells of organisms. So our genes, you can see this yellow region here is what we refer to as a gene. A gene is a segment of DNA that's carrying instructions for one polypeptide, meaning one protein. So one polypeptide chain or a protein. So the gene is going to determine what the organism is like, its appearance, how it survives, how it behaves in its environment. They give very detailed instructions for a living being to make molecules called proteins. And a gene, this gene here, is going to be determined by the order of nucleotides within DNA. So the instructions in a gene that tell the cell how to make a specific protein, the adenine, cytosine, guanine, and thymine, are basically the letters of that DNA code. They stand for those chemicals, the adenine, cytosine, guanine, and thymine, respectively, and they're going to make up the nucleotide bases of DNA. And the code is going to be determined by the specific order of nitrogenous bases. Again, that A, T, G, and C in that gene. The code is going to consist of 3 sequential bases. So you can see that's kind of indicated here. You have what we term a triplet. That's going to be 3 sequential bases. These bases, 1, 2, 3, or you could go the other way, 1, 2, 3. So it's going to be 3 of those bases that forms what we call a triplet. And each triplet is going to actually code for a particular amino acid, like we're going to see during the process of translation. So again, we have our cell, nucleus. Inside that nucleus is your chromosome of DNA. You have your genes, which are again just segments of DNA carrying the instructions for one polypeptide chain or one protein. And then within that, you can see we have these nucleotide pairs. They're going to match up with that complementary base pairing. And 3 of these together form what we know as a triplet. It's that DNA code. Talking a little more about this genetic code, each DNA molecule consists of many genes, and each gene carries coded information. So each 3-base sequence on DNA, or in other words, that triplet code that I talked about on the previous slide, is going to be represented by a complementary 3-base sequence on the mRNA called a codon. So I'm not sure the word codon is up here. But this table is actually showing you these different codons for mRNA, something we'll talk a little bit more about during the process of transcription. So there are 64 possible codons. And the way we get 64 is we have 4 bases in RNA, right? RNA doesn't have thymine, it has uracil instead. So you have A, U, C, and G. So that's 4 bases. And you have 3 places in a codon, right? It's a triplet. So 4 to the 3rd power equals 64. So we have 64 codons here. Each one of these 3 letters combinations, those are each codons. And 64 codons are going to encode for only 22 amino acids. There are 3 stop codons here in red. We also have a start codon. Methionine is our start codon AUG. So there are only 20 possible amino acids, so this means that some amino acids are going to be represented by more than one codon, right? You can see that here, leucine, you have multiple codons that are going to code for the amino acid leucine. And this redundancy here actually helps to protect against transcription errors. This genetic code here is universal because it is the same among all organisms. So for example, UAC is a codon for the amino acid serine, right? If we look at this, we find UAC, or sorry, I said that wrong. UAC is a codon for the amino acid serine. UGC is a codon for the amino acid cysteine. So codons are going to be written from the 5' end to the 3' end of that mRNA molecule. And the stop codons, UGA, UAA, and UAG, are going to signal for the end of protein synthesis. And something that helps me remember these 3 stop codons right here is UAA is you are away, UAG is you are gone, and UGA is you go away. So that just kind of helps me remember those 3 as being stop codons. And again, you can see your different codons. Many of them are going to be coding for the same amino acid, right? So the question here is, what is the genetic information stored in DNA used for? Anybody know? So it's going to be to direct the synthesis of proteins. Synthesis just means creation. So the genetic information stored in DNA is used to direct the synthesis of many different kinds of proteins that each cell requires. So protein synthesis is important because the proteins created during this process control the activities of the cells. So without these proteins, many of the processes in the body would fail or not work properly. So this slide here shows you an overview of what we're about to cover for protein synthesis. So during protein synthesis, that DNA is going to be copied in the nucleus to make RNA. So we're going to take that DNA, in light gray here you can see that RNA molecule, because DNA can't leave the nucleus. The RNA, this mRNA, is then going to move to the cytoplasm where it attaches to a ribosome. The ribosome is going to translate the coded information from the RNA to create a protein molecule with a specific sequence of amino acids. The protein is then going to fold into its particular shape. Remember back when we talked about the primary, secondary, tertiary, and quaternary structure of a protein? So each protein has its specific shape that's going to enable its specific function. So let's go ahead and get started by talking about step 1 in protein synthesis, which is transcription. This is a step of protein synthesis where the language of the nucleic acids, that base sequence in DNA, is going to be translated into the language of proteins, that amino acid sequence. So we're going to be starting off first with transcription, right? So transcription is where we're taking that DNA and we're synthesizing mRNA, right, to then be used in the translation process. And here are some of the big ideas that you should be able to explain by the end of this lecture. So you should know what is the meaning of the term transcribe. Why is DNA transcribed? How is it transcribed? How do the ribosomes know when to start transcription and stop it? What's done with that transcribed copy of DNA? What happens during initiation, elongation, and termination, which are the 3 parts of translation? What's the function of mRNA? What is a codon? And how does this relate to tRNA? So these are all questions we're going to answer throughout the rest of this lecture. So to answer one of those questions, first off, it asks you why we have this transcription process. Again, transcription is where we're taking that DNA and transcribing it into mRNA. And the reason why we have to do that is that DNA stays in the nucleus. So we talked about how the information stored in DNA is central to cellular function. So that cell needs to keep the DNA protected in the nucleus. The DNA is also so large that we can't get it out of that nucleus, right? So what we're going to use instead is messenger or mRNA. So transcription is the first step in decoding a cell's genetic information. During transcription, enzymes called RNA polymerase are going to build mRNA molecules that are complementary to a portion of one strand of that DNA double helix. So again, that DNA needs to stay in the nucleus, so we're going to use this mRNA to make this complementary strand here, and then that mRNA is going to be able to go out to the cytoplasm and attach to the ribosome. And I just wanted to bring this up as a review from our biochem lecture. RNA molecules are different from DNA molecules in several important ways. So first of all, RNA is going to be single-stranded, right, rather than our double-stranded DNA. Their sugar component's going to be different. So DNA has a deoxyribose sugar, which you see right here, whereas RNA has a ribose sugar. And then the last major difference that they have is that thymine, that nitrogenous base, is not going to be found in RNA. Instead, it's replaced by uracil. So RNA molecules, also because they are single-stranded, they're not going to form helixes, right? Rather, they're going to fold into complex structures that will be stabilized by internal complementary base pairing. So again, double-stranded, single-stranded, deoxyribose sugar, ribose sugar, thymine versus uracil. The difference is there between RNA and DNA. So now that we know that RNA is different from DNA, what is the role of RNA? So RNA is kind of our go-between molecule that links DNA to proteins. Again, this is because DNA can't leave the nucleus, right? So the RNA is going to copy the DNA code in the nucleus, and then it's able to carry that code into the cytoplasm to the ribosomes. All of our RNA is going to be formed in the nucleus, and that's called RNA synthesis. RNA differs from DNA because, again, uracil is substituted for thymine, and RNA has ribose instead of deoxyribose sugar, and is also only single-stranded. And we have three different types of RNA. We have our messenger RNA, or mRNA, ribosomal RNA, or rRNA, and our transfer RNA, or tRNA. We're going to look at each one of these three types of RNA more in depth on the next slide. So three types of our RNA are shown here. We have our messenger RNA, or mRNA, which you've already heard me talking about this lecture. This is, again, single-stranded, and the code from that DNA template strand is going to be copied with complementary base pairs in the nucleus, and that results in a strand of mRNA. So this is what's formed in the process of transcription. This is formed in the nucleus, and then it moves out into the cytoplasm. We also have our ribosomal RNA, or rRNA. This is the structural component of ribosomes. And again, ribosomes are going to be the organelle where the translation process of protein synthesis occurs. So along with tRNA, it helps to actually translate the message from the mRNA into a protein. And our tRNA, or transfer RNA, is the carrier of the amino acid. So it has a special area that contains a specific triplet code. We term this the anticodon. So our mRNA has the codon, right? Whereas our tRNA is going to have the anticodon. And that allows each tRNA to carry only a specific amino acid. So the anticodon region of that tRNA is going to complementary base pair with the codon from the mRNA at that ribosome. So it's going to be adding that specific amino acid to that growing polypeptide chain, or in other words, our protein. So let's get back to talking about transcription. I think I gave you all the background information that you need to know about RNA in order for transcription or RNA synthesis to make sense. So transcription is the synthesis of mRNA from DNA. So that DNA, you can see your DNA right here, it's going to actually split into 2 strands, very similar to what we saw in DNA replication, right? Only in this case, instead of having a leading and lagging strand, we have our template strand right here, which is going to be used to synthesize the RNA. And then we have this informational or non-template strand up here that is not used for this RNA synthesis process. So transcription is going to go from the 3' end to the 5' end of the template. So if you follow this back here, your 3' end starting here, going to that 5' end. Not really something that you need to understand in depth. This isn't a genetics course. Just something if you're wondering what these mean on each end of the figure, you have a 5' end and you have a 3' end. So transcription forms an mRNA, which you see here in yellow, and that mRNA has a complementary sequence to the template DNA strand. And it has the exact same sequence as the informational strand. So if you look here, thymine pairs with adenine, guanine pairs with cytosine, cytosine pairs with guanine, adenine pairs with uracil. This strand of mRNA should be exact same as this non-template or informational strand, with the exception, obviously, that thymine is in this non-template strand, uracil is going to be in the mRNA strand. So again, that's going to be the difference between your mRNA and this informational or non-template strand. So the genes in this DNA, they are going to be composed of what we term exons and introns. So we have this gene in DNA, and it has exons and introns. Exons are going to be express sequences. So these are the part of the gene that actually codes for amino acids. Whereas the introns are intervening sequences. They are non-coding segments. They're just interspersed amongst the exons. So the production of a functional protein involves the transcription of the gene from this DNA into the RNA. And we need to remove these introns and then splice together the exons, because again, those exons are our express sequences. That's what we want for our protein. So before we do this processing, before we get rid of these introns, this right here is what we call pre-mRNA. So before RNA processing, it's referred to as pre-mRNA. Then the newly formed mRNA is going to be processed. It's going to be edited before translation can actually begin. And this is that RNA processing. You can see these introns are now gone, and we have spliced together our exons, right? So introns are going to be removed by special proteins called spliceosomes, leaving only these exon coding regions. So this is that end product then of your transcription. You have this mRNA. It is complementary to that original DNA template strand. But it is going to be able to leave the nucleus and go out into the cytoplasm for translation to occur. So before we move on to translation, I just want to make sure that transcription makes sense to you. So from the template strand of DNA shown here, I want you to either in your mind or write on a scratch paper or in your notes the mRNA strand and the informational strand. So go ahead and pause this video now and see if you can do that. So in blue here, you see that mRNA, right? G, C, A, T, A, U, right? Because uracil is here instead of thymine. So what you should see is that your mRNA and your informational strand are the exact same, with the exception of uracil is in the mRNA, thymine is in that DNA. So just as a reminder, RNA synthesis or transcription occurs in the nucleus because DNA cannot leave that nucleus. So you have instead You have this DNA here. You're going to have your pre-mRNA molecule. Again, that's going to be complementary to that template strand. Then it goes through RNA processing to remove those introns. Now you have your mRNA that's able to leave the nucleus and go out into the cytoplasm and attach to the ribosome. So after transcription, we have the second stage of protein synthesis, which is translation. This process is going to involve the translation of the information in that mRNA to, or the translation of the information in the mRNA to protein synthesis. It's going to occur in your ribosomes. Each type of RNA is going to play a role in protein synthesis. The mRNA, again, contains that sequence of codons that determines the order of amino acids in the protein. Individual tRNAs are going to bring the specific amino acids to form that polypeptide chain. And then the rRNA contains binding sites that provide the platform on which protein synthesis occurs. So we're going to look at this process of translation next. So again, after that transcription of your DNA to your mRNA is complete, translation, or in other words, the reading of those mRNAs to make proteins begins. Again, this is going to occur outside the nucleus. So you're going to have this mRNA go outside the nucleus into the cytoplasm. So during translation, that mRNA molecule created in the transcription process is going to deliver information from the DNA to the protein building machines. So you've heard me mention that protein synthesis occurs at our ribosomes. What exactly is the ribosome's function? Well, the ribosome is going to coordinate the coupling of mRNA and tRNA. A ribosome is composed of two subunits. You can see that here, a large subunit and a small subunit. So during translation, ribosomal subunits are going to assemble together, kind of like a sandwich on the strand of mRNA. So you can see that here. Here's your strand of your mRNA. You have the big subunit on top, small subunit below that. So they're assemble kind of like a sandwich, and then they proceed to attract the tRNA molecules that are tethered to the amino acids. So these are your tRNAs. The little colored dots on them are your different amino acids. So a long chain of amino acids is going to emerge, right? And what is a chain of amino acids? It's a protein, right? So this protein is going to emerge as the ribosome decodes this mRNA sequence into a polypeptide or a new protein. So I've mentioned tRNA a little bit, but what exactly is the role of tRNA? So tRNA is going to bind a specific amino acid at one end of it. So that's going to be the stem region. You can see right here, this is the stem region. That's where that amino acid is going to bond. On the other end, it's going to have the anticodon region. This is that triplet code, and that triplet code actually determines which amino acid will be bound up here at the stem. So an example here, tRNA with the anticodon UAU will only be able to load a methionine amino acid to its stem region. So once the amino acid is actually loaded onto this tRNA, it's now called an amino acyl tRNA. And I put down here this little figure to kind of hopefully help it make sense to you. So you have your mRNA, right, that was transcribed in the nucleus, and it has that codon region. So say the codon is UCA. The anticodon of tRNA is going to be complementary to that mRNA. So U matches with A, C and G, A and U, right? Complementary there. And then again, that tRNA is going to code for a specific amino acid right here. So that's what's going to allow us to start with that mRNA. The codons and anticodons match up. that gives us our specific amino acid to make our proteins. So molecules of tRNA are responsible for matching amino acids with the appropriate codons in mRNA. So each tRNA, again, is going to have two distinct ends, one which binds to a specific amino acid, and the other which binds to the corresponding mRNA codon. So if you look here for this process of translation, it occurs in 3 different phases that require ATP, protein factors, and enzymes. So we have initiation, or the start of the process, elongation, where we're making that long protein, then termination, where we stop making the protein. We're going to look at these 3 phases a little more on the next slide. So the first part of translation is going to be initiation. And initiation begins with that mRNA binding to the ribosome. So you see that happening right here. This is initiation. Here's your mRNA in pink. It's going to bind to this blue ribosome, right? Then we have a tRNA is going to come in. That's this kind of yellow thing right here. That tRNA is going to bring your first amino acid in purple here to the binding site. Initiation is always going to begin with that start codon, AUG, or methionine. So that's going to be your initiation process. We had that first tRNA come in here. The codon of the mRNA is bound with the anticodon of the tRNA. We have our first amino acid. Then we have elongation. So elongation proceeds as that next tRNA molecule comes in. So this next yellow one here, it delivers the next amino acid, and a peptide bond actually forms between these 2 amino acids here. So this is going to keep on going on. That first tRNA that was bound here, you can see that AA1 was the first one here. That tRNA is no longer needed. It's going to dissociate, and we're just going to keep on going, right? So each one of these tRNAs is gone as well too. As we keep going, this tRNA is going to go away, that tRNA is going to go away. What we're left is with these amino acids, right, that are bound together by that peptide bond to form the protein. So translation, this elongation part of it's just going to keep on continuing until we hit a stop codon. Remember I mentioned there are 3 different stop codons, UAA, UAG, and UGA. So when one of those 3 is reached, this is what we call termination. So you can see UAG is right here. That tells us, hey, we're done building this protein, and that completed protein here in purple is going to be released, and then we'll do a recycling of the translational components to start this all over again. So that is your process of translation. Again, initiation starts with AUG. You can start with that tRNA binding here, your first amino acid. Elongation, you keep on going, forming those peptide bonds between the amino acid. Then when you reach a stop codon, termination occurs. You're going to release that polypeptide or your protein. So here is a summary of protein synthesis, everything we covered here in this lecture. Start out with your DNA molecule, right? And your DNA is made up of different genes that are going to code for your specific proteins. So your DNA is going to be turned into mRNA. It's transcribed into mRNA during the transcription process. Then during translation, you have the codon region of your mRNA matching up with the anticodon region of your tRNA. And each one of those tRNAs, that anticodon, is going to specify for a specific amino acid. So you have your DNA turned into your mRNA, then you have your tRNA that allows you to get to your protein. So complementary base pairing is going to direct the transfer of the genetic information in DNA into this amino acid sequence of proteins. DNA triplets are going to be coded to mRNA codons. mRNA codons are base paired with tRNA anticodons to ensure the proper amino acid sequence. And the anticodon sequence of tRNA is actually identical, if you look here, to your DNA sequence, except that uracil is substituted for thiamine. So UAC, TAC. So that's the only difference there. So this is that process of protein synthesis, starting at DNA, going to your protein. So this is kind of a simplified picture of this. Again, protein synthesis occurs in 2 steps. Your transcription, that DNA information is going to be coded into your mRNA. That pre-mRNA has introns and exons, needs to be spliced to form your mRNA. That mRNA then in translation is going to be decoded to assemble your proteins. So cellular DNA contains instructions for building the various proteins that the cell needs to survive. So in order for a cell to manufacture these proteins, specific genes within its DNA must first be transcribed into mRNA, and then these transcripts must be translated into chains of amino acids, which later are going to fold into fully functional proteins. So hopefully this process of protein synthesis makes sense to you. So I want to end here by talking about why do we need protein synthesis? Proteins are our worker molecules, right? They're necessary for virtually every activity in our bodies. And proteins have many different functions, as you can see here in the slide. So proteins are necessary for our hair and nails, for our muscles, for cellular messaging to take place, for antibodies. They're in our blood, our brain and nerves, enzymes, cellular construction workers. So they do so many different functions within our body. And as a reminder, what determines the protein's function? The structure of that protein, right? So if proteins are not formed correctly during this process of protein synthesis, they will have the wrong structure. Therefore, they will not be able to do their function, right? So that is why protein synthesis is so important. Okay, we are finally at the end of chapter 3. Go ahead, if you haven't already, do that transcription translation activity. And then also you have a quiz over chapter 3. And I hope that you enjoy the rest of your week. And I will see you for our next lecture where we start covering chapter 4. Mahalo.